It's a great pleasure to be introducing this year's Hooke Medal winner. The Hooke Medal is something the BSc has been running for a while now. It's in recognition of a fantastic contribution to the field of cell biology for lab heads who've been running their lab now for about 14 years. And it's a great pleasure because actually looking around the audience, there are quite several Hooke Medal winners from previous years here today. So it's nice to have you all here. I'm sure you'll join me in congratulating this year's winner, who is Karibaan Hodivala Dilke, who, as I said before, everybody knows as Kebs. So Kebs told me that she's going to be giving a sort of historical perspective on her career, which I think will be very interesting for many of you here. So I'm just going to be rather brief in my introduction to her. She was an undergraduate at Southampton, a PhD student with Fiona Watt, and her supervisor of her postdoc was a guy called Richard Hines at MIT, and he was a very wise man. He's given me several bits of invaluable advice over the years, and I think Kebs is going to share some as well. So pay attention. They're actually very important and worth listening to. She returned to the ICRF to a tenure track position, and she is now the Professor of Angiogenesis and Deputy Director at the Barts Cancer Institute. And I won't tell you about her work, because she's going to tell you about that today, but it really is a great pleasure to be introducing her today. Kebs, come on up and get your medal.
I do want to say I'm really thrilled to get this, actually. In particular, because I think it really reflects contribution of all the fantastic people I've worked with. Teachers, mentors, collaborators, and of course everybody in my lab who are really patient with me, all my bad jokes and my craziness. So this is to all of them. So thank you from them. Thank you very much. So as Jordan said, I'm going to give you a little bit of history, because I thought this was kind of a nice opportunity to tell you where I've come from and perhaps what shaped me becoming a biologist. I'm not sure any of us are born a biologist. I don't think any of us know what a biologist really is when we're little, but I think it's something that comes naturally especially to people who do cell biology, and I think that it's kind of intrinsic. So I wanted to tell you kind of where I've come from, what shaped my career, I think it's not over yet, and my current research interests. And actually, to put it in perspective, really where it all started was with art and music. And you might think that that's a bit odd, but that is the truth, and that is where it started. And actually, my first jobs were working in botany, and looking down the microscope at stuff that came out of plants, and then to malaria and other human diseases, and then cancer and blood vessels, which I've really kind of got stuck with. But throughout all of that, the main theme has been working on adhesion, why cells stick to their environment and how that influences biology. So there I am, age 7. I was born to a Parsi family, and born in London, and kind of a strict upbringing. You know, education was very important, but then there was also this weird dichotomy where being creative and being artistic was very important indeed. And my father was an architect, and my mother a graphic designer. And so art was very important, and it really started in the garden where, you know, I used to have to put my coat on, you have to go outside, you have to sit out there, find something and draw it. And this is what my mother especially was very, very keen on teaching me to draw. And I think that's been a really important part of biology for me, because it really taught me to observe properly. And although digital microscopy and confocal, you know, all this stuff is fantastic, I still try and say to my students, you know, look down the microscope and draw what you see. They think I'm completely bonkers. But I think it really helps you to observe, and I think that really helped me. So now, this gorgeous lot of women with a sixth form from my school, and at school actually, the biggest influence was music, and we had a very strong music director, Mrs. Sue Cutler, who's right here. And she really taught me that actually there are no boundaries, right? You can be creative. Whatever it is you do, you can do more of it, and you can do it your way. And she actually pushed me to take up a Saturday school position at the Trinity College of Music, where I was surrounded by amazing musicians who are all 1,000 times better than I could ever be. And it was a fantastic experience about learning about your instrument, learning how to play, obviously, but also learning how to conduct, learning how to write music. And those features together, I think, are something that we probably all share as biologists. Learning, when you run a lab, it's very easy to say, okay, you do that project and you do that project. But actually you find that after a while, people kind of settle into the sort of biology that they're best at. It's the same as having an orchestra, right? You can make a flautist play the violin, but actually the reality is that they'll play the flute much better. And I think trying to realise that in the lab is actually a really, really important feature. Now, another important thing about Mrs. Cutler was that she was married to this man, Mr. Cutler. And when I spoke to her and told her that actually what I really wanted to do was biology, and I didn't really think I could make it in music, she said, well, you need to talk to my husband. And I think she bullied him into giving me a summer job at the Jodrell laboratories at Kew Gardens. And he obviously had no choice because his wife had told him that he had to take me on. And he kind of, said, OK, well, have a go. And it was fantastic there because there, what I was really doing was sent out into the Kew Gardens with a bucket and a test tube and a pair of scissors to collect samples and then bring them back to the lab and analyse them. And this was at a time when with plant biology, Western blot analysis or even sort of normal running gels was kind of a new thing. So there was just sort of told, well, there you go. There's some money and some equipment. Have a go. And it really gave you confidence that you didn't need to follow rules necessarily, that you could actually create your own experiment and think of your own ideas. So those summers at the Jodrell laboratories actually coincided with my BSc degree at Southampton, and this is where cell adhesion really came into it. My undergrad project was supervised by this guy, Peter Thorogood, who very sadly died in a climbing accident in the late 90s. But I remember when he gave lectures, he used to like wearing big scarves and had big pockets. And his pockets were full of different coloured chalks. And the way that he'd give his lectures was not with PowerPoint or slides or anything. He would stand there and draw cross-sections of embryos, beautiful cross-sections of embryos in all of these different chalk colours. And he really brought out, I think, what I call the love of art in biology. So seeing these image competitions now, which I think we all really enjoy, was very important. So he was interested in how tissues talk to each other and how they interact, how they stick to each other, and how they control each other in craniofacial development. And when I left Southampton, I wasn't actually sure that I really wanted to do a PhD. And I started talking to this guy here, John Lanyardo, who some of you might remember worked at the Royal Holloway University, and he was a parasitologist and he worked on schistosomiasis, and his kids went to the same school that I went to. And so we used to go around to his house quite a lot, and you'd go around and the house was, normal house with kids and lots of mess and clothes hanging all over the place, and he'd have the dining table full of these little black and white pictures of these sorts of parasites, looking at their cytoskeleton and getting excited about going to Africa to collect samples. And he made biology seem like this really exotic and exciting opportunity, and convinced my father actually that I shouldn't go and do medicine. What I should do is really do biology, because that's where all of the discovery is really going to happen. So being inspired by my past, being inspired by guys like John Lanyardo, and also by my grandfather, who I never actually met, but was sent to Aden in the 50s by the Brits to try and work on malaria, which was obviously a big problem. He was actually a homeopath, but he eradicated malaria in Aden by throwing quite a lot of DTT around. But it worked for him, and it did get rid of malaria. So with that armament behind me, I decided that I could actually apply for a tech post at Imperial College with these guys, Prof. Sinden and Pete Billingsley, who was a young postdoc at the time working in Prof. Sinden's lab, where we were going to look at the basement membrane changes that occurred during the malarial life cycle in the mosquito. And this is where I really had a lot of fun, partly because right next door to the lab is a pub. And at that time, going to the pub and then coming back to the lab was not frowned upon. Eating and drinking in the lab also didn't seem to be frowned upon either. And so people used to just stay in the lab until the very early hours in the morning and dissecting guts out of mosquitoes and sticking them under the microscope. And so that was a very exciting time where really the real love for biology and discovery came through. And it was Bob that then said, look, if you really want to have a career in science, you do need to go and do a PhD. I said, well, how do I work out where to do a PhD? He said, well, read some papers and see which ones you like best and write to those people. There was no application or anything like that at the time. We just sort of wrote to somebody and saw if it happened. And I happened to write to this particular lady, Fiona Watt, who you probably all know. And she was I don't know how many of you know Fiona, but she's got incredible energy, enthusiasm. She just never gets tired. She knew exactly what all of us were doing in the lab at any time, and she really showed us how to put power into our experiments. And with her, I did a PhD trying to understand how integrins, which were new adhesion molecules at that time, how integrins controlled keratinocyte biology. Now, coming to the end of my PhD, I really wanted to go for a conference at Keystone. It's one of those ski-stone conferences. I'd never skied before, fancied myself as a skier. And so I said to Fiona, I really need to go to this conference on collagen, I think it was. And she said, you can only go if you have some job interviews in the States. And I said, well, I don't really want to work in the States. She said, well, you can't go to the conference then. So I decided to write to a few people, again, not actually applying for any. I don't think I've actually applied for a job in my life, now I think about it. Wrote to these guys and said, okay, I've got a few job interviews lined up. So I did go to the Ski Stone conference. I didn't ski very well. I popped my knee quite badly, came back in bandages. But I did have an interview out there with this guy, Richard Hines. And I ended up working with him for about 5 years. And what Richard really, really impressed me with was how, whatever it was, you should think big, right? And I was talking to Jordan a bit earlier. He is a very even-tempered person. You know, he didn't have these sorts of ups and downs and this excitement and the downs. And everything was, that sounds interesting. Or if something went horribly wrong, oh, that's a pity. So, it was the same tone for everything. And I remember, reaching over his desk at him one time saying, can you get worried about this with me? And he said, no. He said, if it's worth getting worried about, do something about it, and then you don't need to worry about it. If you can't do something about it, don't worry about it. And that was his sort of philosophy. And so I learnt an awful lot there. I used to go into his office with piles of data thinking, right, this time I'm going to impress him. And every time I came out with him saying, that's interesting. And him giving me more and more information. And it was at that time that we developed, in collaboration with others in the States, the beta-3 integrin knockout mouse that that really laid the foundation for coming back to the UK and working on my tenure track position with this guy, Professor Ian Hart. Now, Ian is a fantastic scientist, and he took me a step further from phenotyping in mice and trying to just understand the biology, not just understand, but understand the biology, to really thinking about, okay, well, how does this work in people? How is this going to affect cancer patients? And really trying to say, well, that's interesting, but how are we going to apply it? And I don't think I've got there yet. I think I'm still on that step, trying to work out how we can apply our work that way. But he was a very rigorous scientist. He's retired, but he's still a very rigorous scientist. A true mentor, very critical of your work. He always says, you need to be more critical of your work than anybody else will be. You need to challenge your data before anybody else challenges it. Always aim for perfection. And don't let anybody else be able to say that they've been able to contradict your results. And I think that's been really important to me. He's also taught me how to write grants. He's still helping me write my grants. And so he's been a massive mentor and influence for me. So those are the people that kind of led me to where I am now. And this is what I work on, adhesion and tumour angiogenesis. So angiogenesis, the formation of new blood vessels, is essential, we think, for all tumours, all solid tumours to grow. And based on that, there have been massive efforts and billions of dollars spent on the production of anti-angiogenic agents. Unfortunately, those anti-angiogenic agents aren't really the golden bullet that we thought they would be. So now we've come to a point where we need to think about how we can exploit vascular modulation in other ways to try and treat cancer better. Are blood vessels simply conduits for nutrient supply and oxygen supply, a waste disposal system? And if anti-angiogenic therapy isn't going to be the way forward, then which way do we actually need to go? So what are the problems? There are lots of problems with anti-angiogenic therapy. Limited efficacy. We thought that anti-angiogenic agents were going to work on all tumours all the time, and they don't. These agents, there's evasive resistance. So basically the drugs are given, they work for a little while, and then the tumour outsmarts the drug. It causes an increase in hypoxia. Now, that doesn't take a rocket scientist. If you're taking away the oxygen supply, you decrease oxygen levels, you increase hypoxia. Now, increased hypoxia in the tumour environment is a bad thing. It also reduces the efficacy of certain chemotherapies and radiotherapy because these therapies depend on the oxygen tension being available. There is preclinical data that anti-angiogenic therapies can also increase metastasis. They can actually increase the spread of cancer. And obviously, it can reduce delivery of chemotherapeutics to the cancer. So these are all problems that were facing us. They've been coming up over the last 12 or 15 years. We've been trying to ignore them, but actually the fact is that these agents need a lot more work to make them work better. Now, in our lab, we've taken over the last 12 or 15 years a genetic ablation approach to try and understand tumour angiogenesis. And in particular, we've knocked out several integrins, beta-3 integrin, beta-3 and beta-5, alpha-3 and alpha-6. This was all done in collaboration with international collaborative groups. But knocking these out one by one or in groups ends up actually enhancing the expression or activity of the pro-angiogenic receptor, VEGF receptor 2. And the ultimate result is enhanced tumour angiogenesis. So why did we do those experiments? Well, these integrins, especially beta-3, beta-5, alpha-3, they're all upregulated on blood vessels in tumours. They stick out of the surface of the endothelium, so they'd be good targets to inhibit. And we know that adhesion is important in angiogenesis, to be able to move around and form tubes and be able to actually develop into a blood vessel system that feeds the tumour. We thought we needed those changes in integrins. But these genetic ablation data tell us that actually we don't. And in fact, if you get rid of them, or if you get rid of their function, you might actually enhance angiogenesis. And at the same time that we were doing these sorts of experiments, there were inhibitors to beta-3 integrin, one in particular called selengitide, that was being developed as an anti-cancer agent. And it was only last year that, unfortunately, that drug has come out of clinical trials for not showing any overall benefit. Because although preclinically it was able to inhibit angiogenesis and inhibit tumour growth, in people it didn't seem to work. So it came to about 5 years ago when we hit this kind of scary moment of a crossroads. We'd spent the last 10, 12 years trying to inhibit angiogenesis, trying to block tumour growth. And actually, we hadn't got there at all. In fact, we'd gone the other way. So we needed to think about what we could do. What can we learn from all of those teachers and mentors that we've had so far about being creative, thinking outside of the box, thinking big, and trying to work towards something that might actually work by making careful observations of what we've done already. So I'm going to describe to you 2 short stories, just to give you the flavour of 2 stories that have been coming out of the lab recently. The first is to turn anti-angiogenic therapy completely on its head and think about, well, if getting rid of blood vessels increases hypoxia, if getting rid of blood vessels reduces chemotherapy delivery and reduces chemotherapy efficacy, then maybe what we should do is increase tumour angiogenesis. Heresy. The second one is a slightly separate project, but it's also important because it's about endothelial focal adhesion kinase and how manipulating endothelial focal adhesion kinase doesn't actually have much of an effect on the blood vessels, but it enhances chemosensitization. So let's start with this first story. So, in fact, I remember when I first arrived back from Boston and I was presenting the anti-angiogenic ideas to Ian, he said, well, you know, Kebs, it's not going to work. And I said, but why, you know, everybody's working on anti-angiogenics at the moment, and, you know, it's the best thing to do. He said, well, pancreatic cancer. Look at pancreatic cancer. All right, look at pancreatic cancer. Pancreatic cancer is a very hypovascular disease. It has very, very few blood vessels. And out of all of the cancers, it has the worst prognosis. It's extraordinarily hard to treat. Even surgery doesn't always work. It causes desmoplasia. This is an increase in fibroblasts and collagen deposition, which makes the chemotherapy very difficult to administer. It's very drug resistant. We can't work out why. Epithelial to mesenchymal transition is very high in pancreatic cancer, and metastasis is usually present at diagnosis because people haven't been diagnosed with it properly earlier on. And so current strategies include surgery, which only are effective for it's only applicable for even 10% of pancreatic cancer patients. And cytotoxic chemotherapy includes a whole cocktail of different chemotherapies that can be given. One of them is gemcitabine, a nucleoside analog, but this has very limited benefits. Very limited improvements are achieved. So let's think big. Let's turn it on its head. Let's think about what we really want to happen. What we really want to happen, perhaps, is to increase the tumour blood vessel density. Then we can increase gemcitabine delivery, and we can reduce cancer growth and increase survival. It's just like a little fairy tale, and why shouldn't it work? Well, how are we going to increase tumour angiogenesis? Well, I told you a little bit about selengitide. This is the inhibitor of beta-3 integrin that was actually originally developed as an anti-angiogenic agent. We had an extremely talented postdoc in the lab called Andrew Reynolds, who showed that the reason, possibly, why selengitide didn't work was that when selengitide was delivered to patients, it was delivered at very high doses. But then the patient goes home, comes back 3 days later, and it's delivered again. And in between that, the concentration of gemcitabine in the bloodstream goes right down. So Andy asked, well, what's the effect of low doses of gemcitabine compared to high doses of gemcitabine? And what he showed was actually low doses of gemcitabine have a pro-angiogenic effect. They upregulate VEGF receptor 2, that pro-angiogenic molecule on endothelial cells, and that actually increases angiogenesis. So this then gave us a tool to increase angiogenesis using low-dose selengitide. Now, what else can we throw in to try and increase gemcitabine delivery, make the blood vessels bigger and fatter? Verapamil is a calcium channel blocker that increases vessel dilation and reduces the efflux of nucleoside analogues like gemcitabine out of tumour cells. So it traps the gemcitabine in the tumour cell. And then, of course, we need a cytotoxic chemotherapy, gemcitabine, because that's what's most commonly used in pancreatic cancer patients. So this should improve the delivery and improve cancer control. So we did the experiments. We treated mice either with gemcitabine alone or with our triple combination therapy of low-dose selengitide, verapamil, and gemcitabine. And these are images of ultrasound in living animals of their tumours. And the ultrasound can tell us about blood flow, tumour perfusion, or tumour blood volume. And what it showed us consistently was that when you have this triple combination of low-dose selengitide and verapamil with gemcitabine, that you can increase blood flow in the tumour, either in the core or across the whole tumour, you can increase the perfusion, and you can increase tumour blood volume. So we were delivering more. And in fact, we confirmed that by doing HPLC of gemcitabine from those tumours and showed there was actually more gemcitabine in those tumours. The net effect was that in KPC mice, these are a mouse model of pancreatic ductal adenocarcinoma, that when we treated these mice either with placebo in blue, gemcitabine in green, we actually saw very little effect, very similar to what we see in patients. But with the combination of low-dose selengitide and verapamil, we stopped those tumours from growing. And this is in combination with an enhancement in blood vessel density. What we're looking at here is a cross-section through the tumour, and we can count these blood vessels, and we see an enhancement in blood vessel density. So this was really going against the dogma, going against what we had already worked on for several years, that actually enhancing and not reducing angiogenesis could be a way forward. What about hypoxia? What about this key feature of anti-angiogenic agents that we wanted to try and get rid of? Well, GLUT1 and what does that stand for? It'll come to me. These are markers of hypoxia. We looked at tumour sections and we assessed the level of hypoxia and showed that, in fact, in animals that had been treated with low-dose selengitide and verapamil with gemcitabine, we could reduce hypoxia significantly. That this actually reduced pancreatic cancer progression, the amount of invasion of those pancreatic cancers was reduced significantly, and the number of liver metastases were reduced significantly. Now, we always thought that more blood vessels would increase metastasis because you'd have more segues. But in fact, that's not necessarily true. And overall, we could increase survival of these animals. Low-dose selengitide and verapamil with gemcitabine could actually significantly increase gemcitabine. And note here that we stopped the treatment here at 9 weeks after the beginning of treatment. So actually, if we continue this, and we've done this experiment more recently, we can keep these animals alive for much, much longer. But is there a direct effect on the tumour cells? We were aiming to actually show that there wasn't going to be a direct effect, but to our surprise, there was. So we showed that actually here, looking at intracellular levels of gemcitabine. Now, for those of you who don't know much about gemcitabine, gemcitabine has a sort of whole gemcitabine state, and it also can be metabolized to its triphosphate state when it gets into the cell. So in tumour cells, we showed that verapamil could indeed enhanced the amount of gemcitabine inside tumour cells, and this is what we expected, that had been published before. But interesting, low-dose selengitide also had a similar effect, not to the same extent, and then those two together had an additive effect of trapping gemcitabine inside tumour cells. This is without the angiogenic effect, right? This is in culture. And then interestingly, the triphosphate, the metabolized form of gemcitabine, was also increased in cells treated with low-dose selengitide. And this effect was seen in proliferation assays, where we were able to show that low-dose selengitide had very little effect in helping to kill endothelial cells after treatment with increasing concentrations of gemcitabine. But in all the tumour cell types that we tested, low-dose selengitide actually enhanced the efficacy of gemcitabine. And we went on to show that it's having molecular effects on the molecular modulators, the metabolism modulators of gemcitabine, not via beta-3 integrin, but actually via beta-5. So in summary for this part, we were able to show that low-dose selengitide could enhance tumour angiogenesis, that in combination with verapamil and gemcitabine, this improved tumour blood flow and gemcitabine delivery. It reduced the hypoxia, the desmoplasia I didn't get a chance to show you. It reduced cancer progression and reduced metastasis and enhanced overall survival. And to our surprise, it also has this dual action control, improving gemcitabine metabolism. So what's the future of this dual action therapy? Do we dare take it into clinical trials? Well, the oncologists are chomping at the bit to try and test this. I think there's still a lot of basic research that we should do first, but we'll see how things pan out. What about other cancer types? We're looking into lung cancer and colon cancer at the moment to see if it's also effective in other cancer types. Other types of cancer therapies, other types of chemos, radiotherapy, and possibly for other diseases or even physiological processes. Is there an advantage of enhancing angiogenesis for issues like chronic wound healing, for example? Can we use this as a therapy for other diseases? So the second flavour I wanted to give you was about how we can think differently about tumour blood vessels, not just as delivery agents to try and deliver oxygen and nutrients, but to really control and crosstalk with the tumour cells. So you might remember this slide that I showed you a little earlier, where we showed that knocking down these integrins had this effect on VEGF receptors, and that actually the overall effect was an enhancement in tumour angiogenesis. And we kind of got stuck there until we realised that what we needed to do was move downstream to this molecule, focal adhesion kinase, that signals downstream of both growth factor receptors and integrins. And you'll know that focal adhesion kinase, at least in vitro, is important in focal contact formation, cell migration, actin organisation. It has scaffolding functions, and also is important in the survival and proliferation of cells. And all of these features make it a likely regulator of angiogenesis. So we wanted to test the role of endothelial FAC in tumour angiogenesis. So we developed some inducible endothelial-specific FAC knockout mice using Marcus Frutiger's PDGFB Cree ERT mice. So these are endothelial-specific Crees that after tamoxifen administration will induce Cree activation just in endothelial cells. And we compared non-floxed animals with our FAC-floxed animals. And the experiment I'm going to show you here is where we inoculated the animals with tumour cells first, allowed those tumours to develop, then administered tamoxifen to induce the knockout of focal adhesion kinase just in the endothelium. And we gave them some type of therapy, either DNA-damaging therapies such as doxorubicin or radiation therapy, and we looked at the effect on the growth of those tumours. And to validate the system, we did various biochemical tests, and here's an example of some immunofluorescence looking at the blood vessels in a section of the tumour, either from our wild-type controls or our endothelial-specific FAC knockouts. And you can see that FAC is expressed nicely in that endothelial lining and also in the tumour cells around. But in our endothelial FAC knockouts, we lose most of that endothelial FAC, even though it's still present in the tumour cells, and this is the merged image just showing the loss. And so we were very excited that we were actually going to hit something that could block tumour growth and block angiogenesis. But these were the results. When we compared B16 melanoma tumour growth in the wild types and the knockouts over several days, we saw no significant difference in tumour growth. When we did the same for a carcinoma, saw even less significant tumour growth. So this was kind of heartbreaking, because when we looked at the blood vessels, we realised that there was no change in blood vessel density, there was no change in blood vessel perfusion, there was no change in apoptosis of the endothelium. So all of that work leading up to these experiments, thinking that actually if we can hit focal adhesion kinase because it has all of these reported effects that should be important in tumour angiogenesis, actually in vivo, in a whole organism, have no significant effect. We really hit a brick wall. But at about that time, there were some new ideas about the endothelium and how the endothelium might actually not just be a wall of the blood vessel, but that actually might signal itself to control tumour growth. And these are called angiocrine signals, paracrine signals, also juxtacrine signals that arise from the endothelial lining and can affect the tumour cells around them. And the theory is that upon DNA damage, the endothelium responds by changing its molecular profile, changing its paracrine profile, its secretome, and that can somehow affect the tumour cells immediately around it. And a paper from Michael Heeman's lab from MIT came out about in 2010 that really inspired us to think, well, these sorts of factors are being identified, but what controls those factors has not been identified. So Bernardo Tavora, who was the postdoc in my lab at the time when we were doing these experiments, said, look, let's add doxorubicin to this experiment. Let's see what effect FAC deletion just in the endothelial compartment might have on tumour growth if we did that. So we repeated the experiment that I showed you earlier, injected the animals with the melanoma cells, gave them tamoxifen after the tumours had started to grow to delete endothelial FAC in an established tumour, and then treated them with a DNA-damaging agent. And this is what we saw. That the tumours in the wild-type mice grew basically resistant to the doxorubicin treatment. However, if you just delete focal adhesion kinase in the endothelial compartment, it was sufficient to sensitize those tumours to DNA-damaging agents. And in fact, we showed the same with a carcinoma, that if you inject the carcinoma cells first, then induce endothelial FAC loss and irradiate the animals, similar to a radiation therapy, we could stop those tumours from growing as fast as they do in their wild-type controls. And indeed, when we looked at sections of those tumours, we saw evidence, at least initial evidence, of a paracrine effect. What we're looking at here is tunnel staining in cross-sections of tumours grown in wild-type mice that were treated with PBS or treated with doxorubicin, or in our EC-FAC knockouts treated with PBS or doxorubicin, and then looking at perivascular apoptosis. And you can see that in a wild-type scenario, doxorubicin treatment does increase the apoptosis in the tumour cells, as you might expect it, actually not significantly. But just by losing FAC in the endothelial compartment, FAC expression in the tumour compartment is normal. You can increase apoptosis in the tumour compartment. And we went on to show that proliferation did exactly the opposite, that proliferation of those perivascular tumour cells were reduced. And that this paracrine effect could be recapitulated in vitro. We were able to take conditioned medium for endothelial cells, either wild-type or FAC-null endothelial cells, and then put those onto tumour cells in culture and look at the proliferation, the survival of those tumour cells in MTS assays. And in the absence of any doxorubicin, those cells are not affected. If you take wild-type conditioned medium, at day 3, add doxorubicin, you can see that there is some effect of the doxorubicin on the tumour cell proliferation, but over time resistance occurs and those tumour cells just start to proliferate again. But if you do the same from the FAC knockout conditioned medium, that those tumour cells don't become resistant to the therapy. Now, we know that upstream of a lot of cytokines production, so a main paracrine family cytokines, is controlled by NF-kappa B. And there have been several reports over several years showing that TNF, LPS, mitogens, DNA damaging can stimulate NF-kappa B signalling and can stimulate cytokine production. But the role of FAC in all of this was really not known. So we asked, could DNA damaging therapy affect cytokine production via FAC in the endothelial compartment? And we went on to show that indeed that was the case. That in fact, loss of endothelial FAC was sufficient to reduce NF-kappa B signalling. Here we're looking at endothelial cell NF-kappa B activation. This work was done in collaboration with Neil Perkins in Newcastle. After doxorubicin treatment, wild-type cells will produce a lot of have very active NF-kappa B, whereas that just doesn't happen in the FAC nulls. And when we look at those cells, when we look at subtypes of NF-kappa B, namely the P65 subunit, you can see that after doxorubicin treatment, the P65 subunit of NF-kappa B goes from the cytoplasm into the nucleus, where it becomes activated. Whereas in the FAC knockouts, that simply doesn't happen. And here, when we quantitate it, you can see that that P65 subunit is simply not getting into the nucleus. And the net effect is that the cytokine profiles after doxorubicin treatment in wild-type endothelial cells is upregulated consistently, but in the FAC knockouts is not. So FAC, you could say here, is perhaps acting as a regulator, perhaps even a master regulator of cytokine production from endothelial cells after DNA-damaging therapy. Now, Michael Heeman, that guy from MIT, who first started looking at angiocrine signals, particularly focused on IL-6, and we were able to show that IL-6 was one of our cocktail, and indeed, by ELISA, we were able to verify our protein blot arrays and show that wild-type endothelial cells will produce a lot of IL-6 after doxorubicin treatment, whereas our FAC nulls produced very little. And now, if we give back IL-6, do that rescue experiment, give back IL-6 into those tumours, into the FAC nulls, what we can do is we rescue that phenotype. Now those tumours start to become sensitised to the DNA-damaging agents. So in summary here, the loss of endothelial FAC is actually sufficient to enhance the effects of DNA-damaging therapy. It's not about delivering oxygen, it's not about metastasis, it's not about any of what we thought blood vessels were important for, even up till 5 years ago. But actually that they can signal to the tumour cells and control how the tumour cells respond to chemotherapy. And then the molecular mechanism behind this involves NF-kappa B and the cytokines that are produced. So here, when you give DNA-damaging agents, it affects not only the tumour cells, but it affects the endothelial cells. That when FAC is present, you get high NF-kappa B activation. This causes cytokine transcription of cytokines that actually protect those tumour cells from the DNA-damaging therapy. But when you don't have FAC, those cytokines aren't made. That invisibility blanket is not produced, and now those tumour cells are left naked and susceptible to the DNA damage. So rather than thinking about this stage, 5 years ago or so, as a scary crossroads, I think now we can think about it more as arriving at new avenues, new ways of thinking about tumour blood vessels, new ways about thinking about how to modulate tumour blood vessels to perhaps control cancer better. And I can't leave without acknowledging the people who did this particular set of work. Bernardo was involved in the endothelial FAC project, together with a whole load of people in the lab and from the institute, and Ping Pu Wong, also known as Tony, who's looking for a job and is a fantastic guy, worked on the low-dose project together with our collaborators. I want to make a special thank you To Stephen Robinson, who's now running his own lab at UEA. He was my right-hand man for about 12 years when I first came back from the States, and I'm very pleased that he's running his own group now. And now my new right-hand person is Louise Reynolds, who has also been with me for about 12 years and was a postdoc with me previously. And without them, none of this work would have been possible at all. Of course, my funders, I need to thank them. Please keep funding me. And with that, I'm happy to take any questions.